bit deeper into reproduction, to look at the actual mechanics of how asexual reproduction happens. So when we're looking at asexual reproduction, the mechanism or the mechanics, how does it actually work, really comes through cell division. So the way that a starfish regenerates, the way that it regrows its arm, the way that a plant will produce another copy of itself, the way that fungi reproduce, the way that hydra reproduce, really all comes down to the concept of making more cells, as well as sometimes making new organisms. And so when we look at cell division, we're going to focus in on mitosis, which is the type of asexual cell division. So when we take a second to look at what is cell division, so the what of it, what is happening? What we're actually going to do is take an original cell, which is sometimes called the parent cell. We're going to copy the DNA. We're going to divide the copies. And we're going to produce daughter cells, or offspring. Where we have to be careful is that there are actually two types of cell division. There's mitosis and meiosis. We're focusing, of course, on mitosis first. What we'll see is that cell division should only happen when signaled. So applying cell division and into part of the cell cycle. So trying to understand what is cell division, when will it happen, and then looking at why and how it should happen. This is an example. If we look at this picture, we see kind of these very regular shaped cells. This tells us that it's going to be a plant cell. So we'll look at mitosis happening in certain cells, meiosis happening in certain cells. If we get a little bit deeper into mitosis, mitosis is the form of asexual cell division. So asexual cell division is going to be mitosis. I'm a little bit frozen up, so let me just talk for a second. Mitosis is going to, there are a few key things to know about mitosis. One of them is that mitosis will produce two identical, what we call diploid cells. So this is why when in the last unit we talked about homologous chromosomes in the last section. So if we look at this original cell, we want to get two identical cells as a product. Why do we want this to happen? Well, we want clones or identical cells so that we can grow, so that we can repair. We want the DNA, we want the cells to be genetically identical. So we're going to use mitosis to copy the genome or the DNA, and again, we're just dividing the copies. Right? This idea that we're multiplying by dividing. And so when we look at what is actually happening, we want to look at the original cell. Most of the original cells are going to have chromosomes that come in pairs. So chromosomes in pairs. And these are called homologous. Looks like homologous, but it's called homologous. Homologous chromosomes. Now, we can look at any cell and predict what the offspring will look like. In this example, the chromosome number here is 2. There are 2 chromosomes. But because they are similar size and shape, they're really just one pair. But anytime we have pairs, we're going to call this diploid. And ploid refers to the number of chromosomes. The prefix di refers to 2, meaning we have 2 of each type. And so I count these individually. When I copy these, when I replicate or copy these, I still really only have 2 chromosomes. I count each X, or each doubled chromosome, as just 1. Because remember, a doubled chromosome, the 2 sides are identical. The sister chromatids are identical. So I don't count this as 4. I still just count this as 2. And one of the ways to do this is to count the centromeres. So here would be a centromere region, here would be a centromere region. You can see where it gets skinnier. So if I count the centromeres, it helps me know how many chromosomes are there. So why are we using asexual cell division? Because we're trying to get offspring that are identical to the parents. So I copy the DNA, I divide the copies, and if you look, each of the daughter cells are identical to the parent and to each other. So we know that we use asexual cell division to help create identical cells, but it's really that why, and you also want to ask yourself the how. How does this actually happen? And so that's what we're going to go into next. One of the things before we move on that I really like about this diagram is that it points out to you that this whole thing is not mitosis. Just the dividing of the chromosomes or the division of the nucleus is called mitosis. DNA replication would happen during the S phase of interphase. And then this is going to be called cytokinesis, which we'll talk about in just a second as well. So I do really like this diagram because it does show the concept of your normal cell replicating your DNA, dividing the copies, and then dividing the actual cell and comparing the results to the parent. So moving on, now we want to talk a little bit about the how. So what are the stages of mitosis? This is really how does mitosis occur? It happens in these stages. And so we do want to talk a little bit about the concept of a stage before we move on. When we think of stages, we think of distinct units. In mitosis, they're not really distinct units. They're really more continuous. But if we're looking at a cell, like we're taking a picture, at that moment, we could see the cell stopping in one phase or stage. But the big idea is that this is kind of a continuous pattern. So even though we're going to divide it into stages, we're really going to kind of see that it's more of a flowing event. What we want to notice in this first picture, well, there are a few things that we want to label. This is our relaxed chromatin, and this signals that we are in G1 of interphase. We haven't gotten a signal, the cell is just doing what it needs to do. Another feature that needs to stand out to us a little bit are these centrioles. Centrioles. And centrioles are, we're going to put a star by it, found only in animal cells, even though more recent research has shown they are in other cells other than just animals, but primarily in animal cells. And they're going to help coordinate the spindle. So, if I see centriole, I can assume that this is an animal cell. When I see relaxed DNA, I know it's in G1 of interphase, or GAP1. It's just doing its normal life. At some point, it received a signal to divide. And so what we start to see happen is this division will go through the S,
We'll go through the G2. When you can see chromosomes, this is called prophase. I'm sorry. This is called prophase. And that's when you can visibly see the doubled chromosomes. When you can visibly see doubled chromosomes, that is considered prophase. You also start to see the spindle form. And remember, the spindle are also going to be called your cytoskeleton components. These are proteins that are part of the skeleton or the cytoskeleton of the cell. Next, we're going to have something called prometaphase, and this is probably new to you. Prometaphase, the big idea is that the nucleus breaks down. We need to be able to move these chromosomes around, and so the nucleus will break down. We also see that the spindle attaches to the kinetochore. And we'll talk about the kinetochore and the spindle attachment in just a second. But this concept that these have been divided into two, although sometimes they're seen as just one. Prophase, prometaphase, all of this is kind of happening before we move the chromosomes. So, interphase, prophase, prometaphase. Next we have metaphase. This one's kind of easy to remember because what's going to happen is that the chromosomes are going to line up in the middle. This is also called the equator. And the reason that they're going to go here is this is going to be a checkpoint. We'll talk more about the metaphase checkpoint later. But what's happening is that we're checking to make sure that the spindle is correctly attached. So the spindle will actually bounce back and forth and make sure that there's tension from both sides so that we can actually pull those sister chromatids apart during anaphase. So during anaphase with sister chromatids, so we need to know that word. Sister chromatids are the copies, and they are pulled apart. They are pulled towards the poles. And then finally we're going to have what's called telophase. And telophase is going to be where the nucleus reforms. My pen's not working so well, so I'll say it, you write it, and then we'll move on. Telophase is where the nucleus is going to reform. So we should have singled copies here. We're not quite done. We have done mitosis, but we haven't finished dividing the actual cell yet. We need to take a second before we move on to cytokinesis to talk a little bit about how division gives us two equal cells instead of two half cells. And so what we're going to pay attention to is this theory of the doubled chromosome. What we have here are two sister chromatids, and they are identical. And of course, in a human cell, you would have 46 of these. The DNA is the centromere. The centromere is really the region of DNA where a protein called the kinetochore attaches, and then the spindle fiber attaches to that. So take a second, pause it if you need to kind of talk your way through. Centromere is a region where they're connected. Kinetochore is the protein that attaches to that. And then real quickly, I'm just going to try and throw in that the kinetochore actually digests the microtubule, and that is what pulls them apart. So during anaphase, the kinetochore protein will digest the microtubule or cytoskeleton, and as it digests it, it's pulling apart those copies. The final thing is cytokinesis, the division of the cytoplasm.